Many thanks. So one such prospective study is UK Biobank, but it is just one study in a global ecosystem of bioresources for public health research, which this conference is so nicely illustrating. So if you do have any questions, just pop them into the chat and I'll try and get to them after the talk. So I really want to focus today to give an overview of UK Biobank, which is currently a fairly unique data set in the global ecosystem to enable discovery science. So UK Biobank recruited 500,000 men and women aged 40 to 69 about 15 years ago, between 2006 and 2010, from across Great Britain, across 22 assessment centres located around the country in order to enhance heterogeneity of the cohort. So in different rural, urban areas, different rates of social economic deprivation, and so on. When participants came along to a recruitment centre, we collected comprehensive amounts of data about them on their lifestyle, medical history, environmental exposures, and a whole range of tests related to cognition, hearing, eye tests, and physical measures, blood pressure, lung function, anthropometry, bone density, and so on, as well as a collection of blood, urine, and saliva samples. All participants provided explicit written consent for us to follow up their health over time through linkage to their medical and other health-related records, and to also recontact them again in the future to invite them for further assessments. So early on in the project, we realized that we wanted to study the genetic data, or collect the genetic data of all 500,000 participants in order to enable research into the genetic determinants of disease. And in 2017, we released data from a custom-built genotyping array, which contained 850,000 variants on it, which was then imputed to over 90 million variants. And I think it's safe to say that these data, when they were released in 2017, transformed the use of genetic in epidemiological studies, and it led to GWASs of thousands of phenotypic traits being performed, and many of which are now available online through browsers at the Broad, Stanford, and Edinburgh, and so on. And that then led on to further approaches using genetics, such as polygenic risk scores, Mendelian randomization, and so on. And all of this analysis of the genetic determinants of disease was made possible due to the large size of the study, the fact that we've got standardized assessment of all individuals, and that we have clinical health outcomes through linkage to electronic health records. So from a natural extension of the genome-wide genotyping data was the development of polygenic risk scores. So this is just an example taken from a paper published by the Broad in 2018, which looked at a polygenic risk score for coronary heart disease. And what you can see is that when you take genetic variation across the full genome, each variant on their own contributes very little to individual risk. But when you combine them all together, what you can see is that those individuals who are unlucky enough to be in the top 5th percentile of polygenic risk for heart disease have over a 3-fold risk compared to the general population, and which in fact is equivalent to many monogenic disorders. And it just highlights the importance of common genetic variation in determining very common disease. And genetic risk has often been found to be independent of known lifestyle factors. So while the clinical utility of these polygenic risk scores is yet to be established, a pilot study is currently underway in the UK, which is incorporated into the primary care setting to evaluate the utility of adding a polygenic risk score to predict individuals at high risk of heart disease over and above known risk factors such as age, cholesterol level, blood pressure, and so on, to see whether they may be more suitable for targeted primary prevention measures. So really exciting developments in terms of the translational impact of research based on the data coming out of the resource. So the release of the genome-wide genotype data led to a flurry of excitement about the possibilities of using genetics for health research purposes. And that led to a consortium of pharma companies led by Regeneron to invest in performing whole exome sequencing in all 500,000 participants, which essentially sequence the 2% coding regions of the genome, particularly to identify rare variants that may have functional importance to aid drug discovery. So the first 50,000 participant data was released a couple of years ago, followed by tranches over time, and the data for the full cohort was released in the summer of last year. And this is just one example, which was published by Regeneron last year, which identified rare variants, a rare genetic variant that was associated with a reduction in BMI. It's a variant in a gene that it encodes for appetite regulation in the brain. And this caused great excitement, because what it means is that it can accelerate drug discovery that mimics the function of this loss of function variant in order to produce a therapeutic target for weight gain. The whole exome sequencing data that we've made available is both the raw CRAM data and the joint call data sets processed by a range of different pipelines, depending on the pipeline of choice for researchers. So we're trying to make it as easy to use for researchers as possible. In addition to the whole exome sequencing, a consortium of UK government, pharmaceutical and charities have also came together to perform whole genome sequencing for all 500,000 participants, with the sequencing being done by Decode Genetics in Iceland and the Sanger Institute in the UK. And this will enable researchers to identify very rare genetic variants, both gain and loss of function variants across the full genome, to better understand genomic regulation, the functional impact of structural variation on disease, and to enable us to get a much better understanding of the biological underpinnings of disease. And currently this is the world's largest study to have whole genome sequencing on such a large scale, and is scientifically unparalleled. Again, we're making available both the raw and the joint call data using a variety of pipelines. Data for the first 200,000 was made available to the scientific community at the end of last year, with data for the full cohort to be made available in Q1 of next year. So in addition to the genomic data, we've also focused over the last few years in turning the biological samples that we have, which are limited and depletable, into valuable data that can be used by the global research community. So for example, we've measured biochemistry markers in all 500,000 participants, such as cholesterol and other lipid markers, sex hormones and markers of kidney and liver function, hematological markers in all 500,000, telomere length as a marker of biological aging, also circulating metabolites, which has been performed by Nightingale Health using NMR technology, which essentially measures about 250 circulating lipids and also some amino acids. The data for the first tranche of that is already available. And also, we're now performing proteomics initially for about 57,000 participants, which has been funded again by a consortium of pharmaceutical companies, which is using the Olink platform, initially based on 1,500 proteins, and that's now been extended to 3,000 proteins. And these data will be released in a couple of months' time, with a view to scaling this up to a larger scale, to the full cohort in due course. So UK Biobank's approach has been, where possible, to perform cohort-wide analysis, because this enables a much wider range of research to be performed rather than doing in small case control subsets, and is potentially transformative in terms of the science that these data can support. So this just shows some typical research that's currently being published that uses these data from the biological sample. So you can see here, there's research looking at low white blood cells in relation to Parkinson's, testosterone levels in relation to dementia, meat intake associated with inflammatory markers, in this case CRP levels, telomere length with risk of disease and mortality, and the metabolomics in relation to COVID-19. So as I've said, we've done the metabolites, a pilot study for proteins. We're also conducting a pilot study of transcriptomics, doing single cell RNA sequencing in 5,000 individuals, and with the potential to scale this up over the next few years to larger subsets of the cohort. So each of these areas have now got momentum to have investment, because all of these technologies are hugely expensive when you start to do them on such a large scale. So we hope that in the next 5 years or so that there will be opportunities to perform, for example, untargeted mass spec approaches, as well as more targeted approaches, as well as epigenetics through long read sequencing, and so on and so forth. So we're also enhancing the phenotypic characteristics of the participants by performing imaging study. So we're currently the largest study in the world by some margin to undertake multi-modal imaging in the cohort. And our aim is to recruit up to about 100,000 participants by the end of 2024 to come to a dedicated imaging assessment, of which there are 4 around the UK, to have an MRI of the brain, the heart and the body, carotid ultrasound, and a full body DEXA scan to measure bone density and body fat composition. We've also got funding to do a repeat assessment in 60,000 of those 100,000 individuals in order to enable researchers to assess change in internal physiology and early pathology. And this work's been funded by MRC, Chan Zuckerberg Initiative, and Calico. So these imaging data have been really crucial for the research community, because they enable research into disease mechanisms and disease pathways, how genetic and lifestyle factors influence disease risk through changes in internal physiology, and of course by identifying MRI-based biomarkers of early disease. And they are particular of interest when we have the repeat scans in order to look at changes in brain structure and function related to cognitive decline and early dementia risk. So this is some key research that's already coming out of the imaging data so far. So you can see, you know, ranging from simple descriptive papers looking at the sex differentiation in human brain anatomy, through to the association of cardiometabolic disorders and psychiatric disorders in relation to brain aging, how the structure of the heart and the function of the heart are associated with cardiovascular outcomes based on the cardiac MRI, air pollution in relation to cardiac characteristics based on the MRI, and also how brain structure and function are in relation to mental health and cognitive phenotypes, and how adverse muscle composition based from the abdominal MRI is associated with all-cause mortality. It just gives you a flavour of the types of research that these data can enable. So as Christoph was talking about in the earlier talk, it's really important that for all participants that are included in cohort studies, that we can link to their existing information that's included in clinical healthcare records. So in UK Biobank, we obtain electronic linkage to NHS data sets in all 3 countries. So we have death data for all participants, about 38,000 deaths so far, linked to national cancer registries, about 43,000 incident cases to date. We've also linked to hospital inpatient records, which gives us information on diagnoses and procedures for thousands of cases of many diseases. And we also have primary care data, which gives us information on diagnostic codes, symptoms, signs, referrals, prescriptions, and so on, for just under half of the cohort for general research, and about 80% of the cohort purely for COVID-19 research purposes. So as you can see, because of the large size of the cohort and the fact that we now have about 12 years of follow-up, you can see that there were already, by the end of 2020, there were thousands of incident cases of common conditions for researchers to study. And then over the next 5 to 10 years, even for some of the rarer conditions like Parkinson's and Alzheimer's disease, given their incidence with age, you can see that there will be thousands of cases of disease to really enable robust statistical investigation into their determinants. So just onto the principles of access of UK Biobank. So I think this is really one of the key characteristics of the resource that has enabled it to be such a success over the last few years. And this is because right from its inception, our principles were that the resource could be available to all bona fide researchers, but all types of health-related research that's in the public interest. That's really our key criterion for approving a research project. There is no preferential or exclusive access, so use of the resource is on exactly the same basis for academic and for commercial researchers. The researchers only have to pay for the cost of using it, not for the cost of setting it up. And it's about £9,000 to access all of the data in the resource. So that's the lifestyle data, all the sequencing data, the imaging data, health outcome data, everything, which is about 20 petabytes of data. As I've said, access to the biological samples are limited and depletable, and they're carefully controlled and coordinated. And where possible, we require researchers to perform assays on the full cohort, or at least a very large sample, in order to increase the scientific utility. We do require researchers to publish their findings and return the data back to us. So this is the underlying code used to generate the results and any individual fields that were derived, so that they could be shared with other researchers. So we were opened for business in 2012 to the research community. And you can see that now that we have over 30,000 registered researchers from around the world actively using the resource, and which has increased substantially over the last few years, with about 3/4 of researchers now from outside the UK. We've now got over about 6,500 publications coming from the resource. To be honest, it's quite hard to keep up with what's being published. And also number of citations, number of patents is also exponentially increasing over the last few years. So last year, we released a research analysis platform, which is an online analysis platform in order to further democratize access to the resource, and which brings the research to the data via an online platform, rather than researchers having to download complex, large data sets onto their own servers, thereby making data access and data compute power much more accessible to the research community. So our research analysis platform, otherwise known as the RAP, our RAP is powered by DNA Nexus, stored by Amazon, over 2,000 users, 700 projects underway. And Amazon has also provided free compute for early career and researchers from low middle income countries. So finally, I think UK Biobank, the characteristics of the resource makes it unique because of 4 real dimensions. First, of its large size, 500,000 individuals, the depth of the data that we've collected, lifestyle, genomics, other omic data from the samples, as well as imaging, the maturity of the resource, so now about 12 to 15 years of follow-up with thousands of incident outcomes. But perhaps the most important thing is the accessibility of the data to enable researchers to perform health-related discovery science with the thousands of publications that are now out there. So I just want to say thank you to the generous support of all our core funders, and of course our 500,000 participants who without this study would really not be possible. So thank you very much.